I'm going to give a brief presentation of what we are trying to achieve here is to set up the basis, the concept of a future European research infrastructure dedicated to plant genetic resources. So let me start from the big picture. We all know that plants are essential for life on Earth. They fix all the CO2 that is produced by anthropogenic and geologic activities. And the total carbon in the atmosphere gets recycled every 4 to 5 years. So if photosynthesis was to stop, we would get a total depletion in a very short time. And there are about 400,000 known species of terrestrial plants, and we use about 10% of them as food, medicines, or raw materials for industry, timber, textile fibers, etc. And they are not equally, their biodiversity is not equally distributed on the planet. There are hotspots for biodiversity. The purple regions are hotspots for biodiversity. The the very light regions have very low biodiversity, like Sahara here. And what we know is that this is going to change very rapidly, unfortunately, because in the last report by Kew Gardens, about 40% of plant species were labeled at a risk of extinction at some point in the future. This is a very high proportion. And as you can see, this is the hotspots of extinction risk. And unfortunately, many of these hotspots coincide with hotspots of biodiversity. And for the moment, these are the main reasons for the causes for plant extinction. And as you can see, the main causes for the moment are altered land use, agriculture, etc., etc. It's not yet climate change, which is the fourth or fifth cause of plant extinction. So for the moment, our normal activities without climate change are responsible for most of the extinction. When climate change is going to kick in, this pace is probably going to accelerate. How to prevent this loss? There are 3 ways: in situ conservation in natural habitats, ex situ conservation in gene banks, and what some of us see as the last resort, seed vaults. Now, this is, of course, the most desirable one, preserving the natural habitats and preserving the diversity in situ. But as we have seen, this is getting rapidly eroded. This is presently the most effective and used way of conservation, and allows not only conservation, but also study and distribution for breeding purposes, because all these materials, if they are not distributed, studied, distributed, and used, have reduced value. And seed vaults are, as I said, the last resort. They are just like vaults in which seeds are conserved, but there is no study, no distribution. It's a black box where you just put everything in case of nuclear war, etc., etc. And this has worked in the past. For instance, Icarda was in Aleppo, and most of its collections got destroyed in the Syrian war, and they reestablished themselves in Lebanon. Unfortunately, they had sent the seeds to the global seed vault, and many of them were recovered. And this has recently happened, we don't know to which extent, to the Ukrainian gene bank, which is in eastern Ukraine. So what needs to be done? It's there are two things, in my view. Of course, maintain and preserve genetic resources and attach knowledge for future generations. And in my mind, I compare these to the medieval monks that were in monasteries and transcribed the manuscripts from Aristotle. And that's the way we inherited all this knowledge nowadays. But then, this alone is not sufficient. We need to introduce continuous innovation, which is at the same level of the innovations that were introduced by breeders of previous generations. And here you have a very effective example of a very important innovation that was introduced by breeders at the beginning of last century, which is heterosis in maize. So we need, and we have the tools to introduce innovations of similar importance in modern breeding. So future generations we are going to talk a lot about stakeholders. I think future generations are our main stakeholders here. Now let's come to what is an S3 research infrastructure. So an S3 research infrastructure, S3 research infrastructures are facilities, resources, or services of a unique nature identified by European research communities to conduct and to support top-level research activities. So that's one of the key points of a research infrastructure, top-level research activities, not only conservation and characterization, top-level research activities in their domains. ESFRI selects proposal RIs in strategic areas of research and with an adequate level of maturity to become ESFRI projects, and identifies successfully implemented RIs to become ESFRI landmarks. And then they become a fully developed infrastructure, which is an ERIC, an European Research Infrastructure Consortium. And the calls for entering this roadmap are published every 2 to 3 years. The last one was published in 2021. The next one is expected for 2024, probably. And there are a whole number of these research infrastructures in various domains: physics, humanities, biodiversity, natural sciences, etc., etc. So there are over 30 or 40 infrastructures at different levels of development, but none of them is dedicated to the conservation and improvement of the plants that feed humanity. So there is a very obvious gap in this panorama. And one of the activities of the Progress Project is to identify this gap. I mean, we knew that there was this gap when we started, but identify to which extent this gap is covered by other infrastructures. And then will be a first session this morning to exactly address this point, the complementarities and the possible overlaps of the future research GRACE infrastructure with existing ones. Just to give you an example of how important plant genetic resources are for human history, this is the great depiction of the Great Potato Famine. That was by the arrival of a fungus, Phytophthora infestans, in around the middle of 19th century from the Americas to Europe. So this is the potato domestication center. Potato was introduced to Europe after the so-called discovery of the Americans by Europeans. Phytophthora comes from a different diversity center. So it's not from the diversity center of Solanum tuberosum. It arrived around mid-19th century, and in just 4 or 5 years, it caused a dramatic drop in production, in yield, and as a consequence, in production area. And since many people in poor areas of Northern Europe had potato as a staple food, especially in Ireland, this caused about 1 million deaths and 1 million migrants only in Ireland. So these affected 25% of the total Irish population. That was one of the reasons why you have so many Irish in the US. They migrated. And breeding for Phytophthora infestans is not a make or break thing. It's not a one shot. It's an arms race. Phytophthora is continuously evolving. This is the genetic diversity of different Phytophthora strains. And so you have to find in wild potato species resistance genes and bring them into cultivated potato. That's how Phytophthora resistance was built into cultivated potato to begin with. And since Phytophthora is continuously evolving, you continuously have to find new sources of genes or generate them by some means, by, let's say, modern genomic methods, gene shuffling or something. And it takes, by genetic crosses, by classical breeding, takes 10 to 15 years to get this gene here. By GMO and gene editing, it gets 2 to 3 years, but as we know in Europe, this is not yet accepted widely. So what I want to say is that erosion of the crop wild relative gene pools is depriving breeders of their arsenals to win future arms races, because gene pools of wild relatives of cultivated plants are enormous reservoirs of untapped resilience and resistance to pests. One characteristic of breeding is that it brings a large diversification of morphological characteristics, and Darwin noticed it in the origin of species. So he said, when we look at the individuals of the same variety and a subvariety of our older cultivated plants and animals, one of the first points which strikes us is that generally they differ much more from each other than the individuals of one species and variety in a state of nature. So the wild species has much less morphological variation than their derived cultivated species. This is example from Canis, Canis lupus and Canis familiaris. This is example from tomato. This is Pimpinellifolium, the ancestral one. This is cultivated tomato. So there's domestication brings about a great variation in aspect and morphological characteristics, and more recently also in resistance to pests. But this variation, so the increase in, this is tomato again, you can see the increase in morphological diversity. This is accompanied by a strong decrease in genetic diversity. And this is brought about by two main drivers. One is genetic bottlenecks. So when you domesticate, very often you domesticate a subset of the initial population, so you bring along only few alleles. And the second driver is that while you select for a certain trait, you actually select for a subset of the possible alleles at that locus. So there is further selection at the loci that are under selection by genomic sweeps. So usually a large diversification in say, morphological diversity in tomatoes accompanied by initially a tremendous drop in genetic diversity. And then in modern times, that's an interesting observation. In some species, this genetic diversity started going up again. What happened? what happened was the introgression breeding, so the introgression of genes from wild species that increase the genetic diversity in modern species, in modern varieties. So what's the vision? What's our vision? And this is a slide that I hope will be subjected to criticism, because we have to have our goals very clear when we submit an ESFRI proposal. So the main goals of this adventure will be to characterize this infrastructure, will be to characterize and expand the genetic diversity preserved in situ and in gene banks, and guide future collection and conservation efforts. Then there is a need to homogenize the quality standards for the management of these collections, because right now they are very variable in different countries. We need to provide advanced scientific services and tools facilitating the use of plant genetic resources by scientists and breeders, both for fundamental science. Remember, a research infrastructure is also about research, and for breeding new, more nutrition and resilient varieties. We need to provide training of to all members of the stakeholder chain on the value and utilization of PGRs. And since the international regulations for exchange of biological materials are becoming more strict every year, we need to develop sustainable and equitable solutions for sustainable, safe, and equitable solutions for the exchange of PGRs, while respecting international conventions, phytosanitary regulations, and promoting sharing of benefits deriving from their use. We need to establish a pan-European phytosanitary surveillance systems against pests and invasive species. That's a key thing. We can see the impact of new invasive species and pests on European agriculture. And we need to collaborate on the above objectives with other worldwide initiatives on plant genetic resources. So that's the vision. Are we at point zero? We are not. We have a European cooperative program on plant genetic resources that is alive and kicking. And we have a database of plant genetic resources stored in ex situ, and on the UNESCO database you can find over 2 million accessions of croplands and their wild relatives in about 400 institutes from 43 member countries. And there are a lot of EU projects, both in Horizon 2020 and in Horizon Europe, that have dealt with the characterization of specific plant families, like cereals, solanaceae, legumes. And we'll hear about these in the second session of this morning. So in the development of a research infrastructure, we are here. We are at the concept development phase. There are many, many phases that need to be accomplished before we become a mature infrastructure, and then before maybe sometime in the future the infrastructure is terminated. Everything that starts has sooner or later to have an end. This is the project that deals with this concept development. It started in 1st January 23. It's the organizer of this meeting. And it has about 21 full partners and about 10 associated partners from different European countries, and it's divided into different work packages: inventory and information system, quality certified ex situ and in situ management, technology and scientific services, evaluation and valorization, the RI concept, social and regulatory aspects, governance and financial plan. and communication dissemination exploitation. And the rest of the sessions of these workshops will be dedicated to each of these themes, presenting the blueprint that we have in mind and asking for feedback. And thank you very much for the attention. So any questions or comments from the room or from online? From online, would you please use the chat, which is the easiest way? We see this. Oh, sorry about this. They seem to see the slides, but not in full screen, but in presenter mode. Do you still see any questions or comments? Can you introduce? Do we have one moment?
I'm a partner of the consortium here for Progress, and I'm the director of an NGO based in Switzerland. It's called Terrara. And I saw what we need in the future, and your list, Giovanni. And I was. Yes, and I got a little bit the feeling that many points are lacking here, and you are very much concentrated on the scientific community. And when I read the title, so we want to build up a European community for plant genetic resources, I think we need a much broader stakeholder list here, and not only the science. I know that there are many movements now at the moment going on to bring science on the top of everything that has to rule the next century. But I think we should also see who we want to introduce into this community as well. So we need much more than only have scientists with us. We need to bring all your ideas into the farms, and there we need much more diversity in the farms and on the place. Otherwise, we will be sticking there, and there will be no evolution going on outside the scientific and the laboratories. So I think we should discuss that in the future more, with whom and how we want to collaborate outside of only the scientific community. So I didn't read farmers, for example, in all your presentation.
Yes, I take your point. Farmers were indeed not mentioned. I spoke about scientists and breeders. It is true that farmers are part of the stakeholder group. The general public is part of the stakeholder group there. So there's a large number of stakeholders. That includes also farmers. Whether the farmers should be actively part of the research infrastructure itself, or they should be stakeholders, so they should be recipients of the activities. That's another matter. As you know, there is discussion within this group on the role of farmers. Some that think that farmers are prime conservers of diversity. Some others see farmers as basically recipients of the diversity that is kept elsewhere. So I take absolutely your point. They are certainly stakeholders. The role in the research infrastructure has to be clarified because, as you know, there are different views. Any other questions or comments? Nigel, do you have any comment on this point? Microphone.
So yesterday we discussed this, Bella. I know you weren't here, but it was brought up in the discussion yesterday about farmers being key partners in the project. And I think Giovanni is saying that maybe it's difficult to include individual farmers, but we should definitely have farmers representatives or networks of farmers representatives included in the project. The other group that you didn't mention as being key stakeholders that are, maybe you did mention, but maybe I missed it, sorry, were breeders.
Breeders I mentioned.
Themselves. And so we need to make sure we cover all the bases in this. It's not meant to be exclusive. Let me be certain about that. So, and your voice will be heard. So I think that's key.
Okay. Any more questions from the floor or from the online? The role of farmers in PGR characterization, such as participatory plant breeding context that was raised and answered partially. And I don't see any other question on the chat. So I think we can move to the next session.